very happy to introduce David as well, our second recruit in the last year. David's made major impact in two very disparate areas of protein biology. He started off after his undergraduate degree at the University of Kansas. He joined my lab actually at Washington University, and at the time we were thinking that the tau protein could have essential properties as a prion, but we didn't really have any clean data on that. And what David did during the course of his graduate work was essentially establish, at least from my perspective, that the tau protein can act like a prion, meaning it can form unique structures in cells that are stably propagated in cells. They're transmissible between cells. They can create unique transmissible forms of neuropathology in animals. And this idea is really central now to all of the major neurodegenerative diseases associated with protein amyloids, from Parkinson's to ALS to Alzheimer's. But that wasn't all, because in the course of David's work, he became interested in an emerging field of condensate formation. So he did his postdoc in Cliff Brangwen's lab at Princeton. For those of you who don't know, Cliff is one of the originators of the sort of modern era of condensate studies with his initial observations, and is a major player in this space. But what I think the field has needed, honestly, is to move to an understanding of why condensates form, how they're regulated, and what their biological consequences are. And David addressed this question as a postdoc by studying, basically developing a logic, if you will, for why condensates form, and using basically a simple model based on the idea of capping proteins, nodes and linker proteins. You can basically build a model to control condensate formation in cells and thereby ask very interesting biological questions. And so we're really happy to have David in the center, because he's actually combining two areas that are very relevant to neurodegenerative diseases. And I'll let you go ahead.
Okay, I guess Mark gave my talk for me, so I'm just going to sit down. Is that fine? Thanks for the kind introduction. We'll see how well these slides work. Hi, I'm David Sanders, and I'm Assistant Professor of Molecular Biology in the Center for Alzheimer's and Neurodegenerative Diseases. And my lab is interested in how molecular networks of protein and RNA control cellular states, and how related cellular pathways are hijacked by diseases of aging. Collectively, our research informs how both physiological liquids and pathological solids form according to the interactions of their components. Now, the story that I'm going to share with you began about 15 years ago when I was in Mark's lab. And at the onset of my scientific career, I was fascinated by the vast phenotypic diversity that is observed in the neurodegenerative tauopathies. Despite each disease being caused by a single aggregated protein, tau, they feature characteristic inclusions in distinct regions of the brain, and this gives rise to unique diseases. However, the mechanism that underlied this fascinating observation was unclear when we started our work. And using a simple cell culture model, we are capable of isolating and amplifying unique tau aggregate structures that stably replicated through living systems with remarkable fidelity. And this work has now informed a structural classification scheme for the neurodegenerative tauopathies. Now, at the end of my PhD, I was inspired by an observation. We'd observed that toxic tau aggregates in dividing cells sequester critical proteins for a membraneless organelle called nuclear speckles in the cytoplasm of cells. Now, not only were the proteins mislocalized, but also the properties of these membraneless organelles were disrupted. Instead of featuring a very dynamic state, they had turned into these arrested solids. Now, work from a number of other labs at about the same time had showed that indeed membraneless organelles are prone to being disrupted in diseases of aging, such as neuromuscular and neurodegenerative diseases, and also being hijacked by viral infections. However, to design new therapies that might target this pathobiology, we must first understand the mechanism by which they assemble and encode their unique functions. So I joined the lab of Cliff Brangwyn, and if the slides advance, hopefully it will, because it struggles this one with this one. So I joined Cliff's lab, who had uncovered a foundational mechanism for organizing the cell in the absence of membranes. And along with others, including Mike Rosen here at UT Southwestern, he has shown that threshold concentrations, biomolecules are capable of condensing into a liquid-like state. And we now refer to these membraneless organelles as biomolecular condensates. A couple examples from Cliff's pioneering work are shown here, such as P granules dripping off the nucleus and nucleoli fusing in the developing frog oocyte. So we now understand that the eukaryotic cell is embodied by a number of different liquid-like condensates, each of which features dozens of unique biomolecules, including proteins and RNAs. And while certain membraneless organelles or condensates have clear functions, such as the nucleoli, which produces ribosomes, the function of most remains unclear and contentious. And I've submitted that understanding the mechanism of formation and specificity will inform not only their role in biology, but also how they are disrupted in diseases of aging so that we might develop new treatments. However, to really get at this in the context of living systems, we've had to develop new techniques for bioengineering them from minimal components. So when I was thinking about the ideal system in which to study this, I was inspired by these interesting liquid-like organelles that form in the cytoplasm of cells upon translational arrest. And the reason why I chose stress granules and P-bodies to inform a universal model for assembly and composition was that we know their components very well. They're multi-component systems. All their composition has been explained by very good studies. Moreover, there are the essential proteins for their formation can be knocked out, which allows other components or synthetic facsimiles to be added back one by one. And we have no problems with toxicity here. And moreover, we also know that sometimes these condensates are disrupted in neuromuscular and neurodegenerative diseases, which might provide therapeutic opportunities in the future. Third, they form in a context-dependent manner, which allows the experimenter complete control over their assembly to study how they form. In brief, in non-stress cells, mRNAs are typically coded by ribosomes and other binding partners. However, upon translational arrest, the ribosomes run off the end of mRNAs, and this leaves them naked. It effectively leads to an influx of stripped RNAs into the cytoplasm, and the result are these curious stress granules that have P-bodies that are stuck to their surface. But why it is that one protein goes to a stress granule or one RNA goes to a P-body was completely unclear. We didn't know why it was that they like to stick to each other in this way. So I studied this problem using knockout cells and then adding back synthetic chimeras of the essential protein components. And very briefly, we primarily made use of a nice light-activated optogenetic system to form oligomeric platforms of RNA binding domains in cells. Now, if you have a dimer of an RNA binding domain, you get no condensation. Stress granules do not form. However, if, for example, you have a 10-mer of RNA binding domains, you get this nice dependence, nice formation of stress granules as mRNA floods into the system. And this isn't shown, but P-bodies are appended to their surface. Now, what my work collectively showed was that biomolecular condensates are best understood by breaking them down into molecular wiring diagrams. And from a rich literature on patchy colloids material science, we know that a certain degree of connectivity or valence is required to form a condensed network. This specific degree of connectivity is 3 or greater. And we refer to such biomolecules that embody such multivalence as nodes. Now, there are other biomolecules that are capable of forming linear chains, but in and of themselves, they are not able to form a condensed network. We refer to these as bridges. And finally, other molecules that lack additional connectivity are capable of limiting the degree of networking in the system, and we refer to these proteins as caps. Now, in the examples that I showed you in stress, the network is of sufficient degree that you get condensation upon polysome disassembly. However, what I observed was that if we increase the stoichiometry of these peptide caps, you would get complete dissolution of stress granules and they could not form, although you would still get the formation of p-bodies. Now, what was critical to the predictive power of this network model was the protein nodes folded oligomerization domain, where multiple proteins compete at a single binding site for control over the molecular network. And the winner of this molecular tug of war not only determines whether or not a condensate forms, but also the biomolecules it contains and potentially the reactions it performs. In a series of exhaustive biochemical studies and in-cell reconstitution experiments, we showed that the cell features a multitude of multimeric protein complexes that compete with respect to their composition. Now, proteins that tend to be in complexes that primarily interact with RNA really like to be in stress granules. However, with the complexes where protein-protein interaction networks predominate, they prefer to be in p-bodies. Now, what's critical for the p-bodies and the stress granules sticking together and defining their final composition are shared nodes. And these are ones that are wanting to interact with the essential nodes of stress granules as well as the essential nodes of p-bodies. And they act more or less as a molecular glue that holds them together. However, as I said earlier, since we're using these quantitative live cell reconstitution approaches, we can have complete control over the stoichiometry. And in the case of the typical cells I showed you, the connectivity between stress granule and p-body networks is relatively weak, and you get these liquid-like p-bodies stuck to the stress granule surface. However, if you increase the amount of molecular glue, you cause the networks to collapse into a single entity, a chimeric stress granule and p-body. However, if you knock that out, you get rid of it, then now they no longer can connect to each other and they remain distinct. So, as I mentioned at the beginning of my talk, I ultimately want to take basic biological mechanisms and apply it back to disease. What does this mean for those disrupted nuclear speckles that I described at the beginning in the context of tau aggregation? So in brief, what I did was took a lot of the same engineering approaches to deconstruct nuclear speckles, and I was able to design a peptide cap and engineer it further where we could disassemble the pathogenic tau inclusions while leaving the physiological nuclear speckles intact. And in this slide, all we're showing in yellow are the tau aggregates, and in red is this cap that disassembles the inclusions. So the significance of this for disease remains to be determined. It's one of the things that my lab is currently working on. So we have now made great strides in understanding the formation, the composition of biomolecular condensates. That's all great and well. However, what I was saying at the beginning is that the function of most remains completely unknown. And we do not know anything about how function is hijacked by neurodegenerative diseases. We've seen a lot of correlations, but causation has been difficult to determine. So my vision for my research lab is to mechanistically bridge form, function, and pathology to inform new therapies for disease. And using approaches that go from in test tube reconstitution through proteomics, genomics, and up into the organismal level, we are trying to answer 3 fundamental questions. One is how biomolecular condensates maintain mRNA homeostasis. And in the case of stress granules, we believe that they're acting as a protective solvent to prevent mRNAs from becoming hopelessly entangled. And this is what ultimately leads to the protein aggregation pathology in diseases of TDP43 aggregation. We are also studying dysfunction. How do failures in RNA homeostasis trigger such pathology, such as the aberrant stress granules that are observed in multisystem proteopathy and in ALS? And then finally, using some of our proteomic technologies where we deconstruct individual condensates, we are asking whether network wiring informs drugs that restore homeostasis. And with that, I'd like to thank the people that have been brave enough to join my new research program. We have grown quickly to a size of 7 since joining in November. We are still seeking the new grad student rotation students, so please reach out if you want to learn more. And with that, if I have any time, I can answer a question. Thank you.